For analyzing RNA-Seq data, the first thing we want to do is to load the library, and we're going to show the package HR, and we're going to load it in. And all you need to do is just to put your cursor at the line and then click Run. And it loaded the HR library and also loaded the limma library required by HR. And then we're going to load our data set, which is called x.rda. And we load it in by clicking the Run button. And now we can take a look at the example data set. And by looking at the first few lines of this data set, and we can use this command called head. And then you can see, run this head x. And then you see the data set has 6 columns. And it has those numbers of counts for each gene, which is each row, and at each sample, which is each column. And we want to take a look at the dimension of the data set. We can use the command of dimension, DIM, and then the data set. And then we can see that our example data set has 18,551 rows, which means 18,551 genes, and 6 columns, indicating 6 samples. The first thing we want to do is to generate a vector. We call it counts. That indicates those 6 samples, which condition each sample is from. Here we have 2 conditions. The U, 3 samples are from the U condition, and then the other 3 are from the T treatment condition. And the U stands for untreated. And we run this line. We can look at the counts. This vector we just generated, it just has 6 elements of characters, and this matches to the 6 samples in our data. So the first 3 samples are from U, and then the next 3 samples are from the T. So these need to line up really well. And then now we are ready to use the HR functions. And the first thing we're going to do is to make the DEG list. And this is the object used in HR. And then so I'm just going to run this line. You can see the inputs are our counts and also our conditions group. We run this line, and then you can see what is generated. Okay, we can look at what we produced, and by looking at the names of this object, and then we see it just simply combined our counts, inputs, the counts and samples together in this D object. And then we can type in D, and then you can see that it has the counts as a field, and also another field is the samples that has the samples 1 to 6, and then your groups in the U and the T. The library size is the sum of each column in your input data set. And then you can see the normalization factor here is all 1. We haven't normalized yet. Okay, the next thing is to normalize. And we're going to run this line for normalization. is called calculate normalization factors. And the method we're going to use is the TMM. And this is the default method in HR. And now we look at D again, and then to see what has been added. And then you can see that the normalization factor now, they're not 1 anymore. So it has been calculated for each sample, the normalization factor, using the TMM method. So next thing to do is to estimate the dispersion. So we're going to estimate a common dispersion first. And then we look at the D again. Notice that I always use this D and do a lot of things to it. So every time I do something, I'll look at it to see what has been changed. And now you see a lot of things have been added. So the first thing is still our input counts, and then those are the samples. That's what we had. And now we have a common dispersion number here. It's one number for all the genes. And then we also added pseudo counts and a pseudo library size and average log CPM. So the next thing we're going to do is to estimate the tag-wise dispersion. Tag-wise means per gene, for one dispersion value for each gene. And then we run this line. And then we can take a look at what was added to the D. And then you can see we added a tag-wise dispersion, and there are 18,546 of them, one for each gene. So now we have done the normalization estimation of the dispersion parameters, and then we're ready for doing some statistical tests. The first thing we want to do is just to compare the two groups. Remember the U and the T, the two groups. And then we're going to run a simple exact test here. And so to look at the results, you can look at the fields using the names of these test results. And then you can see it contains a field of table and comparison and genes. Okay, start again. We can take a look at the fields of these test results, and we run the names. And you can see that we have, it contains a table and comparison and genes. And HR has a very nice function called top tags, and it will rank the genes based on the significance value and the p-value and FDR of your results, and rank your genes. And if we run top tags on our results, test 1, and you will see that all the genes now are ranked based on their significance. The top, the most significant one, gene 16840, is ranked on top. That's the most significant one. And it has give you the log fold change. This is log base 2. And then log CPM, this is a copy per million, and which gives you a sense of how highly expressed this gene, each gene is. The next column is the p-value, and then the FDR. And you notice that those FDRs are super small, so I wouldn't take those negative 110 too seriously. However, the ranks are important. So that is one simple method for testing the difference between two groups a simple design, two-group design. For the next section of the code, we're going to look at a setup for more complex designs, still using this simple example, but we have some more complex steps and targeted for more complex designs of experiments. Now we're going to look at a different way to test for the difference between the two groups and using generalized linear model setting. And to use the generalized linear model setting in HR, the first thing to do is to make a design. What we use is the conditions we set up, the counts vector, and we use the function called model.matrix, and to make this design object. We run this code, and then we can look at what's in this design object. And then you see that this design has a design matrix that contains two columns. The first column is the intercept, all ones for 6 samples. And then the second column is the condition U, and U condition is assigned to a 1, and then the T condition is as 0. So this is the design matrix. So after we make the design, and we're going to regenerate the DEG lists and using similar code as we discussed before, except for when we do the dispersion, the tag-wise dispersion, we need to include the design in the inputs. And that is the line 72 to 79. And we're going to run them. And now we are ready to fit the generalized linear model. And then that's called a GLM fit. And then you can see the inputs are our D2 object and the design. And we run that, and we can look at what's in it. And then we can see this has all the parameters, estimate the coefficients, the design also included, and those. And after we fit the model, we can do the likelihood ratio test for inference. And then we're going to do the generalized linear model likelihood ratio test on fit 2 that we just generated. And then we can look at what's inside by typing the name of the resulted object. And then you can see we have a table that has a lot of information we like. Also carry out the design, the samples, and other parameters. To find the most significant genes, again, we can use the top tags and then use the resulted object test2.lrt, and then we want top 10 genes, and then we run that line. And then we can see we got the most significant genes out, and the numbers will be not exactly the same as your exact test, but the orders should be roughly similar. And if you find the orders of the gene are dramatically different from what you got before, and you need to go back to check your code to look for bugs. That is a generalized linear model way to compare your two groups of the example data. And that wraps up the second chunk of our analysis. Okay, instead of using the negative binomial distribution and generalized linear model, for analyzing the RNA-Seq data, and we can transform the data and then make it fit for a linear model. And so we're going to try the limma-voom package, and the library it needs is the limma, which is a linear model-based analysis that was used for microarray data analysis. And we loaded the limma library. And now we're going to redo our design step and make it according to limma's requirement for making design matrix. So now, and I'm generating this design called design 3 still using the model matrix. And you can see the inputs now looks different from what we did with the generalized linear model in HR. And then we can run this code and look at what is generated. And then you can see now the design matrix has 2 columns, but it looks very different from what we generated for the generalized linear model. Now, it doesn't have an intercept anymore. It just has the 2 conditions, the T and then the U column. And in order to use limma, the linear model, we have to transform the data using this voom function. And then you can see the inputs are our D, this is what we generated in HR, and the counts. And then the design is our design matrix we just generated. And then we can get the plot. And we're going to do that. And then we see that this variance plot, so the y-axis is the square root of standard deviation that represents the variance. And then the x-axis is log 2, the counts plus 0.5 that represents the gene expression. You can see the variance is not the same for genes at different expression levels. And for the low expressors, they have higher variance. So the goal is to make them the same. And that's what voom does. And voom will stabilize the variance. And now we can run linear model. We're going to fit a linear model. And based on our design, the weights are what we generated based on from the voom function. And then you run this line. And you got your results from fitting the model. You can look at what they are. They have the coefficients and design and everything. For limma, to use limma, in order to compare 2 groups, you have to make these contrasts. And if we want to contrast the T condition versus the U condition, and this is how we make the contrast matrix. And then you can see make contrast, and the contrast is condition T minus condition U. The levels we're working with are cons T, cons U. And these 2 names correspond to what you generated in the design. So, and then you can look at this design contrast matrix, and then you can see the T is 1 and U is -1. So what you're getting is T minus U. So now, and we can fit the contrast. And test for significance for this contrast. And what you use is the empirical base. And now you can look at your results in test 3 and to see whether the most significant genes are similar to what you got before. So the top tag, and I didn't specify the number of genes, top genes I want to see, and usually they will send out 10 of them. And then you can see the order is slightly different, but very similar. And now you see the p-values and the FDRs are still very small, but not as extreme as what you got from the exact test. That is fine because they are different tests and they have different assumptions, and you will get different p-values, but the key is that those p-values and FDRs are still very small and the orders are roughly the same. That's the important thing. So, okay, that's all I want to show about how to analyze RNA-Seq data using HR and limma-voom. These are the simple steps, the backbone of those packages. However, the packages have way more things you can try. I strongly recommend you read the tutorial and follow it through to see what else you can do. And plus, you can draw for visualization and other functions you can try. And the important thing for using HR and some other statistical package too is that you understand what method they're using and how to construct their design matrix. Also know how to find whether a design matrix is right or wrong. That's the key.